Dr. Frank Harrell is a professor of biostatistics at the Vanderbilt University Medical Center, where he's also the founding department chair. And so many of us are familiar with Frank's contribution to statistical literature, but specifically here, he's going to talk about Bayesian methods in clinical trials. So thank you.
Thanks for the intro. You know, it's such a wonderful pleasure to work with Jonathan at Vanderbilt. And also, I was glad to hear him mention Bayesian sequential designs, which is what I want to talk about. They're particularly simple and particularly efficient. And what I want to try to motivate to you is that they're very, very clinical, because you can build in various clinical considerations in a Bayesian adaptive design. And there's really two secrets to efficiency and clinical relevance that I'm going to highlight here. One secret is having a sequential design with infinitely many looks at the data. And the other one is having an honest effect size in the design. And so that's something that's not a Bayesian idea or frequency idea, but we know there's this thing called sample size samba, where people are playing in this dance of figuring an effect size that it's affordable to study instead of a real effect size. So you'll see that I have a design loop related to that that tries to ensure that you have an honest effect size while you're doing the other aspects of the design. So there's various considerations in designing a clinical trial. First of all, what are the possible conclusions you would like to draw? Of course, there's all the things that are clinical, like who's the population and what treatment. I'm ignoring all that. But some of the conclusions that you might want to draw are that a treatment is efficacious. There's more than trivial efficacy, which is something I'm going to emphasize a lot. There's inefficacy, actual harm, non-inferiority, similarity, or estimation. So sometimes we just want to estimate an effect without making a judgment. That's often the case in a dose finding or dose response study, but it can be the case in a regular treatment comparison too. What we really want to avoid at all costs is the single most common outcome of clinical trials in the history of clinical trials. And that is, you get to the end of the trial and you don't know anymore after the trouble has been made and you've put patients at risk than you knew before the first patient was randomized. That is actually occurring in more than 50% of all clinical trials. And so having the maximum sample size expended and you still don't have a conclusion is what we really, really want to avoid. And let me mention, by way of background too, the sort of things I'm talking about have major benefits for very large trials. When you get into rare diseases, the benefits are even greater. So when you have a rare disease, you know, you have a lot of trouble randomizing enough people. And most rare disease studies are futile. You could do a futility analysis and it's futile before the first patient's randomized. You could stop for futility. That's really a shame. But one of the shames is you have a small universe of patients and you keep the trial running too long to find out the treatment didn't work. And that means these patients are locked up on a treatment when they should have been released to try something else that might work. So that's related to all of this. What are the operating characteristics? Well, in a frequentist design, the operating characteristic that is given the most play is alpha, which is what I call the type 1 assertion probability, because it has nothing to do with making errors. It's not an error probability, but people call it that. It's just a probability of asserting an effect. And so you could say that alpha is the probability of making a conclusion. Whereas in the Bayesian world, we're not interested in the probability of making a conclusion. We're interested in the probability that the conclusion is correct. So the Bayesian operating characteristics are really emphasizing the correctness of the conclusions that you're making. That is all important. We don't really care about the trigger rate where you fire a conclusion, like what alpha is doing. We want to know about the proportion of times that when you make conclusion X, that X was the correct conclusion. So that's going to drive everything we do. And so another operating characteristic is going to be the Bayesian power, and there's many ways to define that. This is an oversimplified way, is to say that we have a high probability of detecting a minimally clinically important difference if that's really the true effect. And that probability needs to be high. And what we mean is, say, the posterior probability of efficacy is greater than 0.95. We want the probability of the probability being greater than 0.95 to be high, which is one definition of Bayesian power, when you compute it at the average sample size, not the maximum achievable sample size, but at the average sample size, which I'll say more about. Another operating characteristic is what's the expected stopping time. So what's the expected study duration or number of patients you would need to randomize? And you might have another operating characteristic, which is the precision of the estimated effect. And so what you're going to see in the design that I'm going to suggest to you, this design simultaneously solves a number of problems. The design is going to say that you're willing to not care to estimate the effectiveness of a treatment that's not effective. So it's going to give up on having good confidence intervals or uncertainty intervals if you have high probability that treatment is not effective, and you're only going to care about estimating how effective the treatment is when it's actually effective. So when the treatment's working in the right direction, we're going to put a clause in our algorithm that says we need to estimate how effective the treatment is with a certain precision. So here are the challenges that we face. Understanding what an operating characteristic is for Bayesian could not be more different than an operating characteristic for a frequentist design. So the only operating characteristic that's important for a Bayesian is the related to post-data decision making. Are you making the right decisions or not? That's all we care about, other than things like precision sometimes. This is really big. We need to quit allowing gaming of effect size used to choose the sample size. If you're willing to stop very early for futility, you can make major time savings, cost savings. You cannot hang up patients with rare diseases for too long into a futile study if you're willing to do this. If you're willing to stop early for similarity, that will affect the design a little bit, not nearly as much as this. And then we need to formalize the minimum precision criterion for estimating how effective a treatment is. And then we need to be very specific and objective about what we mean by a trivial treatment effect. This is kind of a new consideration for people that are used to p-values, because that's just testing for the existence of a signal, a non-zero treatment effect. But I'm going to suggest that we need to be putting emphasis on demonstrating the treatment effect is not trivial. And when you're willing to do that, when you're doing a futility analysis, this makes it much more often that you'll stop early for futility. Now, the reason all of this is so important is that I would wager $100 against anyone that of all of the studies that end up so-called negative, at least 50% of them could have been stopped very, very early. There's no reason to keep these studies going that end up negative, and a Bayesian sequential design will reveal that to you. So before getting into this example, I want to go to a specific Bayesian design, and there's a lot of material in this link you're going to get about Bayesian operating characteristics and various other things. But this is the design that I'm going to show you. So we're going to let delta be the true treatment effect that you're estimating. So this is going to be some unknown value that you would like to know. This could be a difference in systolic blood pressure means. And the example I'm going to run is for a continuous outcome where you're really looking at a difference in means. We're going to define the MCID to be smaller delta. And then we're going to define this sort of new quantity, which is the triviality threshold. So this is something you might call a minimum detectable delta. It's not the minimum clinically important difference. It's more like the minimum detectable difference, which is a threshold for a non-trivial treatment effect. Usually I would set gamma to be 1/3 of delta or 1/2 of delta. So you take the MCID and sort of divide it by 3 or 2. That is a good sort of starting point for the clinicians to start coming up with the triviality threshold for their treatment effect. And then you need to define a similarity interval for delta, which you might use as minus 1/2 of the MCID to plus 1/2 the MCID. That would be a reasonable similarity interval. All of these are judgments that I made. In a real clinical trial, you would have the clinicians weighing in big time on this. N is going to be the average sample size used for resource planning, which is an estimate of the ultimate sample size based on assuming the true treatment effect is the MCID, although you'll do a better job by putting uncertainty on the MCID, like a prior distribution. And let's say we want Bayesian power to be 0.9. That means we achieve a 0.9 probability at a certain sample size that the probability of efficacy exceeds 0.95. And we're going to use a two-component rule that says we have a very high probability of efficacy in the right direction. So we're actually, say, reducing blood pressure. That probability needs to be really large. And the probability that the improvement is more than trivial is not quite, doesn't have to be quite as high, but it's 0.85. So I'm going to require both of these to be true. So I've got a pretty good chance the treatment effect is not trivial and a really good chance that it's in the right direction. And then there's a lot of other stuff you can say about this, but this is what the design is. So you start off, this is one of your keys to success, is choosing a high information patient outcome variable. So you want to choose the outcome variable Y that has maximum resolution. So you want to have minimum measurement error. You want to have maximum retest reliability. And you want this variable to be a continuous variable if humanly possible to do so. So a continuous variable will have maximum resolution, meaning maximum Bayesian and frequentist power. Then you pick, you elicit the effect sizes. This is the MCID. This is the trivial treatment effect threshold from experts and patients who are unaware of budget limits. And this unaware is something that needs to be in boldface to cut out this game we're constantly playing. And then you compute the average sample size and minimum efficacy sample size N sub P. So this N sub P is going to be the minimum sample size that will give you sufficient margin of error in estimating the amount of treatment effectiveness. And then this N is more for the Bayesian power. And then when you compute this value of N, is it feasible? Well, if it's not feasible, you need to ask the question, did we push hard enough on getting a high information outcome variable? So if you can achieve a higher resolution outcome measure, you can go back up here and start over. This is something you should have done before, but you sort of forgot to do it. So now we're going to dump the original outcome variable. We're going to substitute one with higher resolution, and we're going to elicit the effect sizes that we want to detect for that one. So we're starting over. If you can't develop a higher resolution outcome measure, you need to stop. Now, this is very bold, very unusual to tell a researcher the best thing for your career is to never launch this study. You're going to save money, you're going to save embarrassment. The researcher will come back and say, I know I'm going to be lucky this time. And you tell the researcher, well, of 1,000 clinical trials, very few of them got lucky like you're supposing you'll be. And so we see trial after trial after trial, especially small single investigator trials, launched on a wing and a prayer. And that is not ethical. And if you talk to anybody who's been doing clinical trials a long time, and I talk to senior leaders at FDA about this, that is just not ethical. We need to abandon that idea and just not allow that anymore. So we have this loop here to make sure that you've got a real MCID and you've got an outcome variable for which it has so much information in it that you can achieve your goals. If you're doing a longitudinal study, you can make the information go up even higher. So if you have a high resolution outcome variable that you can measure monthly for 2 years, your effective sample size is going to be much more than the number of patients in the study. Now, once the N is feasible, you can begin the study and do frequent sequential analysis. It doesn't matter whether you schedule these or not. And in the Bayesian world, there's absolutely no multiplicity adjustment for sequential testing. So you can do this after every patient if you wanted to. Depends on how up-to-date your REDCap database is. And the first question you ask, what's the probability that the unknown treatment effect is greater than gamma, which is the point of triviality? So this is an inefficacy consideration. If you have a very low chance of non-trivial efficacy, so that probability is less than 0.025, you're going to stop for inefficacy. So the fact that you're testing this not against zero, but against gamma means you're going to stop earlier. So what happens with a lot of futility analyses is you keep the study going longer, and at the end, the treatment effect is trivial anyway. So this is going to say that's not good enough. If we can't have evidence of a greater than trivial treatment effect, we're going to stop very early. Now, if this is not very low, so you have a hope of having non-trivial efficacy, you ask the question, have you followed at least N sub P patients? This is going to be the minimum number of patients that will give you adequate precision for estimating the treatment effect. If you have not already seen that many patients, so if that's no, you're going to accrue more participants, and then you're going to go back up here and look for, with the new participants, the evidence for inefficacy. Now, if you have accrued enough patients to estimate the efficacy, if the efficacy is likely, now you do your main assessment for whether you have enough information to conclude efficacy. So you already know at this point you have enough precision for estimating the efficacy. If it exists, the question is, does it exist? So is the probability of the treatment effect being greater than zero very high, and the probability of being greater than a trivial effect fairly high? So if that is not the case, you ask, is the treatment effect in the similarity interval with high probability? If that's true, you stop with the conclusion that the two treatments are similar. That's a conclusion we never get with a frequentist design. Otherwise, we ask, are the resources exhausted? And if they're exhausted, we stop with the inability to reach any conclusions. This is what we want to avoid at all costs. This is a waste, but that's a possible end result. And if the resources are not exhausted, we go and accrue more patients and go back up here again. But if this is high, and this is pretty high, we can now stop for a conclusion of non-trivial efficacy. So that's, of course, what everybody hopes for. So now let's actually simulate this design in 10,000 randomized trials, and just to show what are its Bayesian operating characteristics. So I'm going to go back up here. So the way this design played out, we're comparing two means, and we're not doing covariate adjustment like we really should. So we're making it just simple. And if you do a frequentist power calculation, for a frequentist power of 0.9, it required 234 total patients in comparing. This is a two-group parallel group randomized trial. A Bayesian power of 0.9 for concluding any efficacy required 196 patients. A Bayesian power of 0.9 for concluding, for having a high probability of detecting non-trivial efficacy requires more patients. And so that to reach a 0.9 probability of a posterior probability being high for this has to require more than that. The average sample size for a continuous monitoring Bayesian design in this design was 113. So sometimes you'll stop early for efficacy, sometimes for inefficacy, sometimes for similarity, sometimes you don't stop. If you average that over 10,000 trials, the average stopping was at a sample size of 113. Now, the minimum N for estimating precision is 60, so you would never stop before 60 for these reasons. You would only stop with less than 60 patients for reasons of inefficacy. And then we set the maximum N allowable to be 750. So first question you ask yourself, how do you know you're doing a Bayesian simulation and not a traditional frequentist simulation? The first thing that tells you that is no two trials that you simulate will have the same treatment effect. So you simulate 10,000 trials, you'll have 10,000 different values of delta. So the Bayesian goal is to uncover delta that generated the data, no matter what that delta was. So we're not giving any special favoritism to a delta of the MCID or delta of 0. But then when you're simulating this, you have to describe a universe of possible treatment effects. And if that universe of possible treatment effects is exactly the prior distribution you use in your final analysis, everything will work beautifully, it'll work as advertised, and it'll be optimal. But when we're doing these simulations, we would choose this universe to not be really optimal. In other words, it may be disagreeing with the prior distribution we're using in our analysis. And so that's how we show that the Bayesian operating characteristics are going to be excellent, even if the situation that you're analyzing here is not ideal for the Bayesian approach. Now, as you probably know from the arguments that have gone on for a long time in statistics about Bayesian versus frequentist, in the frequentist world, you have a lot of arguments, endless arguments, one-tailed, two-tailed tests, Bone-Ferroni inequality. How do you do group sequential test? How do you get a p-value from a group sequential test? You cannot get one. It's impossible. You can just set alpha. Multiplicity adjustments are just constant arguments in the frequentist world. In the Bayesian world, you have one argument, which is the prior. So you're focusing all this mess into one big argument, and then you specify your prior that you're going to use in the analysis. And for the purpose of simulating operating characteristics, we may have a prior in the analysis that doesn't agree with this universe of treatment effects that we used in simulating the trial. So that's how you don't stack the deck in favor of the Bayesian analysis when you're doing your simulations. So we're going to have sequential assessments with unlimited looks at the data, and we can stop at any time for inefficacy with no minimum sample size. Now, there's a side detour that we need to take here that relates to things Jonathan talked about, which is if you're not doing a platform trial and you're going to stop as soon as you have evidence for inefficacy, you've hired a bunch of research coordinators, research nurses, and everyone else. You have this whole clinical trial machine. How are you going to pay them if you stop the study after 3 months and it was going to be a 3-year study? If you have a platform trial, you have a natural way to move on and pay your staff. If you're not doing a platform trial, this gets to be a problem with staffing and people's livelihoods. Now, what happened in this particular simulation? We simulated 10,000 trials under a universe of treatment effects under certain constraints, and half of those trials stopped early for inefficacy. This is a huge thing. So if we use 10,000 different values of capital delta, and half the time we were able to stop early with a conclusion of inefficacy. The average sample size at which we stopped for a conclusion that the drug doesn't work was 62 patients. Remember, the study was powered to need like 220 to 250 or so patients. But what are the operating characteristics? Once you're a Bayesian, you have to think about using different denominators. So in a frequentist world, a denominator would be all of the trials in which the treatment effect was 0. how often did you say there was an effect? So that's alpha. So that has absolutely nothing to do with decision making and nothing to do with Bayes. So the Bayesian operating characteristic would be, of all of the times when you stopped early for inefficacy, what proportion of the times were you correct? So that is the all-important appropriateness of the decision criterion. So we stopped early for inefficacy 5,184 times. Of those times, 5,020 of those, the decision we reached was correct in that 5,020 of those trials were actually generated by a treatment effect that was trivial or worse. And so our accuracy, our Bayesian operating characteristic is 0.97. So of all the times that we stopped early for inefficacy, 97% of the time, that was the correct decision. Can't ask for much better than that. What about the times we stop for similarity? It's much harder to stop for similarity. So 634 trials were stopped early with the conclusion that the treatments have similar outcomes. The average sample size at which you stop for that similarity is pretty big because it's so hard to stop early for that. Of these 634, 607 of them were the correct decision. That's 96% accuracy. That's your Bayesian operating characteristic. Now, how often did we never stop and we went all the way to 750? This is what our design is meant to prevent. So in 172 out of 10,000 simulated trials, we went all the way, and our conclusion was we don't know anything. We have no conclusion. We just know that we spent the money. We've randomized 750 patients, which is the maximum we were going to randomize. Now, if you look at what was going on in these 172 clinical trials, and you know this number is tiny compared to the way clinical trials are actually designed nowadays, this is a very good outcome. But what was going on when that was the outcome? Well, the median unknown treatment effect was exactly equal to gamma. In other words, the clinical trials for which we're unable to reach a decision is when the actual treatment effect is right exactly at the point of triviality of the treatment effect, gamma. So it makes sense that if your unknown efficacy is right at the threshold for triviality, you should have maximum trouble trying to stop that trial for any kind of conclusion. That's exactly what happened. Now, we can stop at any time when n is greater than n sub p. N sub p is 60, I believe, for non-trivial efficacy. So how often did we stop with a conclusion of non-trivial efficacy? We stopped 4,010 trials out of 10,000. The average sample size at which we were able to estimate the treatment effect and have a high probability that the treatment is effective. In other words, the probability is greater than 0.95 that the treatment improved patients, and it was greater than 0.85 that it improved them more than gamma. We were able to achieve that with an average sample size of 102, which is less than what was in the power calculation. And then what are our operating characteristics for that from the Bayesian standpoint? Thinking of your denominator is now all the trials in which you concluded a non-trivial efficacy. So your denominator is 4010. Of those 4010, how many of those was your conclusion of efficacy correct? So that was correct in 3,913 times. So out of 4010 that we stopped early for efficacy, almost all of those, we made the correct decision. Our accuracy is 98% accurate. That's just a fantastic operating characteristic. And if you go into the details of these notes, you'll see what is this under various conditions where your prior is not favorable for the simulations you're running. And you'll see how this varies. And it may go down a little bit, 0.89 or something here. but it never gets really bad. And so, so making a conclusion that the treatment benefits patients, you're almost always correct when you make that conclusion. And it makes no difference that you have hundreds of looks at the data. It doesn't alter how this works. Now, what is the correctness of a conclusion that the treatment not only works in the right direction, but it's better than a trivial effect? So that's a little bit harder to make that conclusion, but we see that we're still 91% accurate when we make a conclusion of non-trivial efficacy. So looking back here at all of these operating characteristics, this is all we really care about. And if somebody asked you, what's the alpha? Alpha has no relevance in decision making. Alpha is an assessment related to a situation where any decision you make is wrong. So that's not really telling you about the accuracy of a decision. So the way I like to word that, there's a lot of ways to word it. Asking one to compute type 1 assertion probability alpha for a Bayesian design is like asking a poker player who wins more than $10 million a year to justify his ranking by how often he places bets in games he didn't win. So you can see if you ask to judge a poker player this way, you're using the wrong denominator in your calculation. Another way to say it is, do you want the probability of a positive result when there is nothing? This is alpha. So if delta is equal to 0, the treatment does nothing. What's the probability that you'll have a so-called positive statistical test? That's alpha. So do you want the probability of a positive result when there's nothing, or the probability that a positive result turns out to be nothing? So that is the Bayesian posterior probability of efficacy, which is why these are the operating characteristics that we care about. So I'm sure I've ticked off somebody and hopefully generated some discussion. So let's see what we have.
Yeah, I don't see any questions yet. Well, first of all, thank you very much. This was very, very interesting and for sure very helpful. And for those especially who care to go this route, which seems to be a good way of thinking about designing trials.
I have two questions,
Yeah, I was going to go to you, Dan,
Frank,
actually. Yes.
into our private chat, and they may be questions that others have. Could you give us a little further definition on gaming the effect size? And could you also tell us a little bit about, you gave us a mathematical definition of a trivial effect size, but could you give us a little more color on triviality?
Yeah, and we have this international discussion board called datamethods.org, which is where it's a discussion board for methodologists to discuss things with subject matter experts. For example, clinical investigators with epidemiologists, statisticians, health services researchers. And so we have a topic on that exact question to try to define various notions besides the minimum clinically important difference. And so that topic on the discussion board might have something for you to of interest. But the main thing that's going on there is that if you calculate the Bayesian posterior probability that the treatment effect is better than the MCID, that doesn't really answer the question. That's what some people do. But let's suppose you observed a treatment effect that was exactly at the MCID. The Bayesian probability of exceeding the MCID is going to be about a half. So you would have a very unimpressive probability of exceeding the MCID when the observed treatment effect, let's say in a very large sample, is right at the MCID. So you don't really calculate it that way. So what we need is something more like half the MCID. We would like to have a high probability if, say, the point of clinical triviality is the minimum effect that anyone would notice. You know, it could be the blood pressure reduction that is beyond what the measurement error in the blood pressure device has. It could be a symptomatic improvement in the patient that the patient notices, even if the patient says, I wouldn't bother to take a pill every day just to achieve that. I can tell something's going on that's positive, but it's so trivial. You know, I can walk 10 more steps a day. That would be a point of triviality. So if you calculate the posterior probability of being more than trivial in terms of the minimum noticeable treatment effect, it might be something like half the MCID, but the clinicians really need to weigh in on this.
Right, and you gave us some bookends there, half or a third. I think that was
Yeah.
very nice. Tell us about N sub P. Is that determined by gamma?
So N sub P is different. And so let's see, where do I talk about that?
I think you had it down where you were showing it.
Yeah, here, so the required minimum precision of the estimate of delta. And so somewhere in here, I defined the actual criteria I used here, and now I'm forgetting where I put it. But basically, you have a little bit of difficulty specifying the precision, but it's going to be something that's more like gamma, but it doesn't have to be gamma. So you might say, I want to be able to nail down the difference in systolic blood pressure to within 3 millimeters of mercury. And so if you had a confidence interval, that would be the confidence interval would have a width of 6. So in the frequentist world, it'd be like half the width of a confidence interval, but we do this in Bayes. So that would be like a Bayesian credible interval. Well, it's half of the width of that. So you have to specify clinically, what is it the clinicians need to know? What is the margin of error of estimating the efficacy that is acceptable? And then we can solve for the n that will achieve that if you have an estimate of the standard deviation of the variable. So it's a usual kind of precision calculation, but using a little bit of a Bayesian flavor.
So it has a relationship to gamma, and that relationship has some flexibility to it.
Yeah, and this could be done independently of gamma, or it could use gamma in the precision estimate. These are conversations that would need to be had that we're not used to having, but you can see from the Bayesian design that when you put all this together, you know, all the parts fit together to create a flow. And you don't have to like any of these parts. You can change any of them. You could change, you know, the intervals for similarity. You could change this 0.95. You could change the order in which you look at things. Although I think this order is pretty good. But these are all things that are very easy to change. And I think you can arrive at a design for a given study that would maximize the clinical relevance while minimizing the expected sample size.
There
Great.
are a couple related
Yeah, we've
questions.
got some good questions now, don't we?
Yeah, and so I guess if I just maybe summarize them the best I can, one, your experience with submitting this type of design to funding, and then these considerations of statistical efficiency versus practical, and sort of what's been the status quo, if I may summarize it in that way.
Yeah, we haven't submitted a design like this for funding, so I'm anxious to get some experience. I do work 1 day a week for the director of drugs at FDA, and so it gives me the opportunity to make a lot of presentations at FDA. And of course, I can't speak anyway for FDA, but there's a lot of interest in this kind of design, especially part of my job at FDA is dealing with rare diseases, and there's a lot of interest in this for rare diseases. So I think there's been some favorable impressions of it, and I'm looking to carry that further.
Yeah, no, judging by the operating characteristics, I mean, definitely looks very impressive, especially with the
let's go a little deeper onto the FDA one for a second. And Ronan Fitzpatrick had several questions there about the threshold of 0.025 for phase 3, and it's having a fixed minimum bar criteria, prevents each trial becoming a sponsor versus regulator fights. And he wanted your comments on rare disease scenarios.
Yeah, I think this is going to be more of a sponsor's choice. You know, anytime a sponsor is willing to declare a drug or treatment ineffective, that's up to the sponsor to be as aggressive as they want doing that. I think if you're doing a frequentist design, this will affect alpha. So there's some different consideration, but that doesn't happen here. So I think there's a good deal of freedom in this. But if you're doing an academic medical center trial, if you're taking infinitely many looks at the data and you want to stop the moment you have strong evidence for inefficacy, so the probability of inefficacy here would be 0.975. You do want to be sort of sure, because you might be giving back grant money. You might be saying, we're going on to the next part of a platform trial and we're going to stop this one. So that's the reason this is as small as it is. In rare diseases, I'm not sure how this would play out and whether that needs to be altered, but I think something between 0.025 and 0.05 is where we'll probably be headed.
Okay. Do we want to open the microphone and make sure that Ronan got his question answered here? Can we do that, Cecilia?
Yes, I can invite Ronan to unmute.
Yep.
You should be able to unmute now.
Yeah, can you hear me?
Yeah.
Yes.
Yeah, so I suppose my point, I suppose it's question / point is the 0.025 alpha constraint. That's obviously kind of the standard assumption for a confirmatory trial. To me, almost seems more like a meta consideration than statistical at this point, where basically you need to just pick a fixed minimum bar. Obviously, that doesn't guarantee you'll get approval, but it's like a minimum bar to even go to the next stage of the conversation. It almost feels like they have that primarily now just to prevent every single approval application being a debate at phase 3. Whereas if you have a Bayesian analysis and these can be picked, based on the science and stuff like that, then every approval application then becomes a regulator versus sponsor fight. And obviously they're very differing incentives. I think that's fair to say. So that's why I probably expect the 0.025 will probably continue to exist, even in the context. I know the Bayesian guidance is coming out next year for confirmatory trials. And I think similarly for like continuous monitoring, that's more of an issue where they don't want operational bias, if that's kind of happening in a, on the site level basis, something like that, or in a more regulated trial, just the resource implications of having the IDMC involved in a continuous monitoring trial. So I think even in the COVID-19 trials where they're using SPRT type things, they were like doing it every 20 subjects. So, for me, many of the decisions seem meta considerations rather than statistical considerations in terms of how the regulators are choosing things. And for me, if the Bayesian framework is to kind of replace or become the preferred framework, it almost feels like you'd need to have a degree of consensus about what that fixed criterion could be. And I know there's kind of a Goodhart's law, when a measure becomes a target, it ceases to be a measure problem if you pick like 0.95 as your posterior probability. But I think if you're going to replace 0.025, in practice, that's probably something that would have to happen, in my opinion.
Yeah, there's a lot of good points there. And the best thing would be to not have any thresholds on probabilities, because we've created a mess. But I think sponsors are probably going to want some sort of threshold that's probably going to force it on us. But like you said, it's mainly a non-statistical consideration. I think there is a reason to go ahead and design trials to have as many looks at the data as patients. So if you have 750 patients, you'll go ahead and design it for 750 looks. Just because if you do that, you don't have to schedule any look. You just decide, when's the data clean enough? When have we followed enough people for a whole year? whatever your criteria, and then you look then. So by not having a schedule, because you allowed for infinitely many looks, it just makes everything so simple with the Bayesian world. Now we've skirted the big issue, which is the choice of prior. And so what I used in this example was a skeptical prior distribution. So I'm assuming there's no super relevant data from a similar study and almost the same drug that we could use to get a prior. And so using a skeptical prior for things like first in man studies, or for cases where we just don't know much, we use what we know. What we know in a given study is usually whether the treatment is curative or not. So if the treatment is curative, your prior can be very wide, and it might include an odds ratio of 0, which would be a cure. And if your treatment is meant to be incremental, then have an odds ratio less than 0.7 is going to be very unlikely. So I'm a big advocate, and I talk a lot about this in this document, about using skeptical priors. And then, there's also all this talk in rare diseases about using historical controls, which is a real mess. It's a total mess, and there's a lot of really bad faith research being done in that world.
And I would say that what you're seeing, in phase 1, phase 2, increasingly, is that Bayesian methods are winning out because the stakes of those are mostly for the sponsor rather than the regulator. And that's why, I think the success criterion, that's what matters. That's when the regulator has to have some kind of fixed rule just to prevent gaming, or having, even just if you're picking a success threshold in trial 1 for this sponsor of X, and then it's Y in this trial, unless there's a very good reason for that, like in rare diseases or pediatrics or similar, you're just going to end up in a big mess. So that's why I feel like the FDA is very, very reluctant to kind of get rid of the 0.025. And I feel unless the Bayesian community comes together and says it should be this instead as a fixed value, and I know there's many criticisms that we found whatever that value ended up being selected, it's going to be very difficult to replace the 0.225 wholesale.
I think you're confusing two issues, though. The more I'm listening to you, you're thinking of an alpha of 0.025. This, what we're talking about here, has nothing to do with alpha.
Oh, sorry, I'm talking mostly about alpha in terms of like the success.
Okay, yeah, forget that idea. Yeah,
Oh, yeah, sorry.
I should have caught that when you first started talking. Yeah, this is a totally different subject.
Yeah. But I'm just just in terms of like this Bayesian sequential trials kind of been used, Bayesian methods been used more in phase 3 trials. Specifically, it's just like my point is more just like, many of the considerations seem to be more meta considerations.
Oh,
like,
that's for sure.
Yeah.
That's for sure.
Like the FDA, I think, just needs to have a fixed criterion because they need to have a fixed criterion to prevent basically controversy and debate and fights with sponsors relating to why did this person have why did this sponsor only have to have a posterior probability of 0.95 and we have to have a posterior probability of 0.975 for being greater than the MCID.
And this fits into whether you're going to use a skeptical prior or a favorable prior. I think FDA is going to be very skeptical about favorable priors.
Yeah, no, that's also true. It's just, I think it's just a meta point. And then, similar for the the continuous monitoring is mostly, I think, it's either a resource issue because the IDMC then needs to be involved much more commonly, or it's an I think, to be honest, I always find the type 1 error considerations in terms of continuous monitoring to be less persuasive, or probably what they're less interested in than the operational bias concerns. And it just happened to be the type 1 error is a good excuse to prevent operational bias concerns if you have continuous monitoring. But yeah, anyway, hopefully some of those points, kind of see it from their perspective a little bit, I suppose.
Maybe we should discuss some of Ying's question. So on the lowercase delta, there's no prior about that. That's just a number. And then the prior that was used was a skeptical prior. And all of the R code is included in this chapter. So the whole code is reproducible, and you can alter it, and you can really see what's going on by looking at that R code. And then, does the sample size calculation account for the sequential process? The beauty of Bayes is you don't need to account for stopping rules in any fashion. So having a stopping rule doesn't alter the meaning of a posterior probability. So that makes it really, really easy. So a posterior probability computed at the end of the study has the exact same interpretation as the posterior probability in the middle of the study. So that just makes it so easy. And yes, that capital delta is the thing we had the skeptical prior for.
Thank you.